I'm really excited about sharing this with you right now. But just to those of you who don't know me, I'm a professor here at UCSF, and one of the things I do is I run Project 2 of the 5 projects in the AViD program. And Project 2 is developing antivirals against proteases and RNA-dependent RNA polymerases. And it's a large group of people. This is really team science. And I'd love to talk about some of these other things, but I'm going to be really focusing on the protease part of it. But I wanted to shout out the other co-PIs, Eddie, Matthias, Ursula, Judd, and Brian, that have really made our meetings every 2 weeks really exciting over the past nearly 3 years. And then we've got a developmental research recipient, both Jun Wang at Rutgers and Margot Pinney here at UCSF, about to head to UC Berkeley. And it's a heavy use of the cores. To just make sure that everyone gets acknowledged that I can, throughout the talk, I'll try to shout out the people that have really made this possible. But I really wanted to let the folks online know how important this team science is. It's just been incredibly exciting to work with this group. And the Project 2 leaders are shown over here, and I'll be mentioning them. Brian Shoigit, I'm certain that you know of in terms of his computational efforts. And then, so I'll be talking about that because that's one of the ways we identified some of these leads. And then Adam Renslow, who's just done the heavy lifting on the medicinal chemistry, the structural biology, predominantly by Klim Verba, but also Bob Stroud's been helping with that. Some of the screening that's been done with Michelle Arkin's group. And then Melanie Ott, who's really been driving the virology, along with Chris White, who's part of that virology core. And then all this is under the umbrella of Nevin Krogan, who's really the lead of the AViD Center, the QBC AViD Center. Now, to riff off of what Sarah mentioned at the beginning, this isn't necessary for this group, but maybe it's important for the folks that are supporting us to, these pandemics they've been here from the time immemorial in the sense of documentation all the way back to 430 BC. With some of those initial pandemics. And if you follow this blue line, even though this didn't end up in science in this form, because science doesn't allow you to put breaks in your figures, so there's a kind of a plain vanilla form of this in the science perspective. But this is the one that Brian and I really enjoy making the points in terms of teaching to some of the students when we're presenting this story, that if you look at the impact and the loss of souls over the centuries, it's been really quite remarkable how intervention coming in and in the terms of either vaccines or some type of therapeutic have really tamped this down. And this is a whole story. There are books that have been written about it, but this was one of the points we've tried to make, and a shout out to all of the centers in this little perspective, how getting groups like this together to work on something and putting egos aside to actually try and get something ready for the future pandemics that are going to be occurring is what brings us all together here. And one of the points we make is that if you look at the ones that have been used here, they've pretty much been repurposed. If you look at remdesivir, the one that came up, it was really developed for Ebola. If you look at what I'm going to be talking about with the SARS-CoV-2 target, it was really focused on med chem and structural chemistry that had been done for SARS-CoV-1, and it was sitting on the shelf ready to be used, so it could be repurposed pretty much for SARS-CoV-2. So with that in mind, I'm speaking to the choir here, that trying to get this new chemical matter and to not just end in showing that it works in an animal model, but to move it forward so that it's available in a phase 1 safety trial so it can be on the shelf is one of the things we're trying to reach for. And so this is a target product profile for folks who are saying, well, why are you working on MPro? Because it's already got a compound that works perfectly well. I agree. It's been a fantastic success story that pharma stepped up and got something in record time, but it does have a lot of liabilities. In particular, those of you know about it, or if you have elderly colleagues or family members, many people over 65 can't take it because the drug-drug interactions. It's a regimen of 6 pills. It does have its other limitations. And of course, there's going to be mutations that come up against any molecule, and there's already a lot of mutations that have come up against it. So our target product profile was to have something that was oral that could overcome some of these liabilities. And a lot of other people were doing this as well. And we're excited about this particular molecule, and these are the ones that I'm going to be talking about for the rest of my time. And it's really 2 molecules that we're very excited about because they are quite drug-like, and I'll spend the rest of the time on how we came up with this, but just to give you the punchline right at the very beginning. AVI 4516 is our lead candidate right now, but this one's coming up really, really fast, 4773. And the main difference for this is the halogenation on this benzotriazole ring. But this is a molecule that has quite a number of drug-like properties. It has a uracil core that's shown right here. It has the isoquinoline moiety that's dropping into one of the subsites. The benzotriazole drops into the other subsites, so the S1 and the S2 subsites. So these are all somewhat drug-like, so that we have a molecule that has the potential to make it into humans. Definitely has a gauntlet to get through. But the really surprising thing, and I'll explain how we came across this, is this propargyl group. Now, this propargyl group is a triple bond, an alkene. And those of you who are aware of click chemistry is really introduced for putting other moieties on there. But the propargyl group itself works as a very weak electrophile. Only if it's sitting next to the active site cysteine to work as a latent electrophile do you get reactivity. And that's something that we're extremely excited about in terms of its potency and its specificity. So the numbers you can read here faster than I can speak, but our IC50s are quite good. There are some that are out there that target this enzyme in the picomolar range, but I think these are quite reasonable. What we're quite excited about is the properties of this molecule that are quite quite bioavailable, are quite similar to the ones that we're getting in vitro. Since it has these properties, we're also excited about its potential pan-coronavirus inhibition, and here's its reactivity against MERS. And since it is reversible, you need to do these K-inact over Ki's to look at the on and the and those reactivities. And these are quite modest in terms of reactivity. You can get these up into the 250 to 300,000 per mole per second, but you don't want that to hit every cysteine that's in the proteome. And then this is in particular what's quite special, that in the mouse models, this is in 7 doses. You add these 2 halogenated groups, and you get it down to working in 3 doses. So that's why we're excited about it. Another point that I'll just mention here, it may come up later. This is a 4-step synthesis and a 5-step synthesis. So the potential of getting this into a CMC production where you have to make extremely large amounts of it, that's another reason why Adam Renslow's group, who's developed this, we're really quite excited about how short a synthesis is. So potential's there, although as anyone in this business that's tried to put these things into the clinic knows that there's a lot of things that have to be overcome. So how did this molecule arrive? And that's what I'll spend the rest of the time on, giving you a feeling for this worked now for MPro, but it can work for other ones. And what we've done, because this is now a project 2 compound, so we're pipeline. So we're not just using this pipeline for MPro, we're using it for other targets. But this breaks down in terms of colors and direction what we're doing. It's there in green where it's things are kind of cold. We're just screening, we're looking for hits. All right. And then it goes through validation and eventually heats up when we get into animal models. The way we find our hits is either through in vitro in cellulo screening, high throughput screening, and that's done a lot with Michelle Arkin's group. But in what the story that I'm presenting here, we've really taken advantage of the computational approach with Brian Schoikat's group, where we can screen billions of compounds computationally, and this is something that John Cordera is quite aware of. A lot of groups are using this, and I think this is some of the excitement of the AI potentially revolutionizing how rapidly this sort of thing can be done. And a lot of us are working on that. But this is how we identified some of the initial lead compounds. The initial hits. And then they are validated, and we want to see whether they're not just brick dust and they can actually cross membranes. Then we move to mechanism of action and efficacy in antiviral studies, eventually look at whether there's liabilities in terms of are they really toxic or are they already requiring boosters like nirmatrelvir does with with ritonavir, and then we eventually move into animals. So that's our pipeline, and it's quite robust, and we thank the AViD groups for supporting that. Now, this is putting in perspective about 600 compounds that were generated by either the enamine approach, which is make on-demand compounds that had been identified through a docking program with Brian Schoikat's group, and taking those hits and testing them compared to nirmatrelvir right here. You can see where our initial hits were, 100,000, nearly a million-fold over the potency in terms of nirmatrelvir. And we sat there for quite a long time in terms of just not finding something that was in the right pockets. And this first study was published initially in Protein Science in 2023, and it didn't show efficacy in terms of animal models. But it was at least an important scaffold that came out of it that subsequently, which is the next paper that's about to come out on bioarchives, was showing much greater potency, where we finally broke through, came out of the forest, and then started to really find something that was now comparable to the nirmatrelvir. So it was a series of computational approaches, of a lot of structure-guided medicinal chemistry to eventually lead us. And then we got lucky, to be perfectly honest, we got really lucky with that Propargyl group that gave us that type of potency that I'll be talking about. So that puts in perspective the 3 years that we spent on coming up with those 2 compounds that I showed on the previous slide. A little bit more detail in terms of the chemistry. That initial screening that was done of the billions of compounds that are out there, this was about 862 million that were initially screened. They weren't available initially, but then once they were identified and they looked, and we did a hit picking party with Brian Schoikat's group, then 146 were synthesized in Ukraine and then sent to us. We did the screening and we picked those that were under 200 micromolar. And what came out of this was this urea dihydrouracil core, and with now at least 21 micromolar. So this was getting a little bit better, even though it was on this floppy urea scaffold. And then adding this thiophene group in terms of ordering some of these compounds and docking them initially against the SARS-CoV-2 structure, these were all starting to now get in the finally breaking through the micromolar range. And then Plim Verba's group with Amy Diallo got the structure showing that that dihydro group was really sitting right in the pocket right next to the active site cysteine, and then filling this S1 and S2 pocket with these two side moieties. So that was really a breakthrough for us, but it was still at the micromolar range. Then Adam Renslow's group really took over and then installed this drug-like isoquinoline group to now fill that subsite, and then also to rigidify this to the uracil scaffold. Now, as soon as this was rigidified, we went up, so it wasn't working as well, but it was now a more drug-like molecule with an achiral center here that could now be added on. So imagine this like the center of the octopus, and these are the arms that are reaching out into those subsites. And that's installing the benzyltriazole group, got us down into the 0.3 micromolar, and then came the luck. What was done is that Adam's group was introducing this propargyl group for click chemistry to now explore other subsites. But one thing that I've learned in any drug discovery program, always test your intermediates, because sometimes the intermediates work even better than the final products. And the group was thrilled when it turned out that this propargyl group ends up forming covalency with the active site cysteine. And so Tyler DiTomasi in the group then took this, did denatured mass spectrometry on it, and you get the nice shift that's just due to the addition of this entire group onto that active site cysteine. It did chymotryptic digestion, showing that we got that individual cysteine that was being labeled with that molecule attached to it. So that was extremely exciting for us. And then the next step was to do the KNACT over KI. And as I mentioned, this is a weak electrophile. It's just touching that cysteine, but only touching it when it's bound into the active site by filling that S1 and that S2 pocket. And so that was promising, because if it's too reactive, it's going to hit too many other cysteines in the proteome. So then how long does it stay? Is it locked on there, or does it come off like nirmatrelvir does? Because nirmatrelvir is a covalent inhibitor as well, but the thioimidate is something that eventually comes off in less than 24 hours. So Tyler then took that inhibited MPro, dialyzed it, and then takes it out of dialysis out of either 1 day or after 7 days, and you can see it's on there. It is covalently attached to it, so it's not going to be coming off, leading to other potential hits elsewhere. And then a little bit of mechanism. The next two slides are on the mechanism, because I think this is what really distinguishes this molecule from some of the other things that are out there and might have a lot of benefit to it. That alkyne group has a carbon group that can be either attacked on the internal carbon or on the external carbon, and we believe it's attacking the internal carbon. Why is that important? You'll see in a moment that the way this is actually binding, and once again, getting the crystal structure of this now with Clem and Amy, you can see that it's covalently attaching to the cysteine, and the isoquinoline and the benzyltriazole groups are packing into the S1 and the S2 pockets. Now, is it hitting other cysteines? All right. And so we did it against a panel of all the other types of possible cysteine proteases. And you can see that we're getting very little reactivity of those other cysteines. So that's exciting to us. That weak electrophile is important. And then here's two slides on previous papers that have been done, that we're not the first ones to work with a propargyl group against a cysteine nucleophilic enzyme. And so it was initially done in a kind of a Ben Cravat effort by hanging it onto ubiquitin to go hunting for deubiquitinases. And this is a case where this is a good capture agent for finding dubs. And that propargyl group is very good at reacting with those deubiquitinases. And then the cathepsin K enzyme that's important for osteoporosis and a target in the drug industry, that's also been shown to work. And then there was even a paper where nirmatrelvir, there was one that was hung on it to get it to work for it. Didn't have great drug-like properties in that case, but compared to ours, I think we've overcome some of the liabilities of this one. But just a moment about this reactivity, if you can just allow me to geek out for a second on this one, and that is that cysteine is only reacting when that propargyl group is placed right next to it and it's sitting less than an angstrom away. If you try to react with just a cysteine modifying enzyme, glutathione or something like that, it will not react. It will not react with those other cysteine containing enzymes because it doesn't have the specificity pocket. But the other thing it has is it has an oxyanion hole that's presumably recognizing the tetrahedral intermediate of that alkyne being attacked with the nucleophile. And that's something that I think is quite special and can be quite useful for moving across other coronaviruses that have the cysteine, have the oxyanion hole, and have that S1 and S2 pocket. So then, of course, we've got to look into the toxicity and other potential liabilities of it. So this is a study that was done to look at other things like the hERG channel, various things like that. And it looks like it's really quite reasonable in terms of not hitting those in terms of that receptor panel. And then these melting efforts, the CETSA, and this was with Daniel Swaney from Nevin Krogren's group, it really is not hitting other cysteines in the entire proteome. So we're really quite excited about that. Now, I'm going to skip over this. I have it here, and it will be in this bioRxiv paper that's under review right now. But you can see just in terms of its efflux of moving across and out of membranes, it really looks quite good. So you can look at those, you can read those faster than I can speak. But this we were really thrilled to see. In terms of microsome stability and really interacting with any of the cytochrome P450s, you can see there's very few really red. It's a little bit pink in certain areas across the different CYPs. So this is quite exciting because it bodes well for not having the drug-drug interactions and the need for a booster. Okay, I want to wrap up in terms of the bioavailability of this, which is also really quite remarkable. Wasn't able to be predicted other than choosing drug-like molecules as you go along that medicinal chemistry effort that Adam Renslow drove. But then if you start looking at just oral bioavailability versus IV bioavailability, you can see you IV it, and then after 8 hours, it's cleared. You do it oral through an oral gavage, and we're 26-fold over the EC90. This was a fantastic surprise, all right? This is a highly bioavailable molecule. And then if you look at BAUS, it gets into the tissue concentrations, but I'll save this if there's questions about it. Final point about this before we go to Melanie's antiviral data is that it does seem to be getting into the brain, and this could be a point of discussion regarding tissue distribution, and could this help with the reservoirs that seem to be in the brain that nirmatrelvir is not able to get at. Okay, finally, this is, if we're going to take this seriously, then it has to show efficacy in an animal model. This is not a prophylactic, this is an infection model, and you can see the regimen that's up here that Raul ran in Melanie's group. And looking at the plaque assays and then looking at 2 days post infection, 4 days and 7 days benchmarked against nirmatrelvir, you can see how we're getting full extinguishing both at the 4-day, clearly all the way out at the 7-day. Now, this was our lead candidate, but then carrying 4773 along with it, we got another nice surprise. And that is those 2 fluorine groups, that halogenated benzyltriazole group, really does make a difference. And so this is curing mice now in 3 doses. So this is particularly exciting for us. I want to be cognizant of the time. And I just want to make a call out for Margot Penny, who's helping us, and Taha, who's been helping us look at the resistance profile. We're taking the data from Judd Hallquist's GSED database of known resistant variants, working with the replicon, and then looking at potential other mutations that are coming up by doing HT-MEK mutagenesis on MPro and picking those mutations that could come up in the presence of our inhibitors, and then building those into the replicon so we're not doing gain of function of a live virus that could get out of the lab. And our molecules are looking quite promising. They're a different profile than nirmatrelvir or enceptrelvir, and we're excited about it, and we're digging into it deeply. Very final point is the potential for broad spectrum across the coronaviruses. And this is done initially with the NIH, with some common cold variants, but the MERS really stood out, and we're quite excited about moving this into animals, and that will hopefully be being started this month with Chris White and Melanie Ott's group. So with that, I know I rushed, but it's I'm here to answer any questions. And as I mentioned, this will be online very soon. So I can summarize that I found this particularly interesting uracil core that we're excited about. The covalent latent electrophile, I think, is an advantage. And then filling those subsites, the S1 and the S2 pocket, with drug-like molecules, I think really helps. The broad spectrum, potential for broad spectrum coronavirus specificity without losing potency. And then we're pursuing this brain penetrance as well as in vitro synergy experiments. And with that, I'll hand it over to Frank to moderate any discussions that may have come up. Thank you.